Hi, I'm Hugo Merchant, and I'm going to talk about brain dynamics in the primate audiomotor circuit during isochronous beat perception and entrainment. Beat-based timing refers to the ability to detect the regular pulse or beat in music and to respond synchronously to the beat during dancing and musical ensemble playing. For long, time beat perception and entrainment has been considered specific to human traits. However, recent studies have shown that monkeys have the audiomotor machinery for regular pulse perception and entrainment. We have trained monkeys in a synchronization task that has two important phases, a perception phase and an entrainment phase. During the perception, the monkeys need to place their free hand on a lever and listen or watch to the metronomes for 2 intervals. And then they were trained to tap in synchrony with this brief stimuli separated by a constant interstimulus interval. We used intervals in the range of 450 to 950 milliseconds, and we use visual or auditory metronomes. Here I show the asynchronies in a polar plot, where 0 represents a perfect timing between the stimulus and the response. And you can see that the mean resultant of the asynchronies of the two monkeys were positive for short intervals, but definitively they were negative for long intervals, suggesting that monkeys have all the machinery to predict a regular pulse. Here is the video of a monkey during the synchronization task. See how well the monkey is synchronizing to metronomes that change the tempo from trial to trial. So we have trial-by-trial information about the timing prediction with the asynchronies. We have the timing precision with the temporal variability. We have the timing accuracy with the constant error. And we also can measure the synchronization error correction with the lag 1 autocorrelation of the produced intervals. So the next step is to try to understand how the brain can perceive and entrain to a single pulse. And the hypothesis of the lab is that there is a main core timing network that includes the supplementary and pre-supplementary motor areas in the frontal lobe, as well as the basal ganglia and the motor thalamus. And we have recorded neurons across this network during this task performance. The first thing that we saw is that the global changes in discharge rate of neurons change as a function of the beat tempo. So here I'm showing a neuron that has a preferred interval for shorter durations, but of course at the population level we saw neurons that were tuned to all the tested intervals. We also saw neurons that were tuned to the serial order elements of our task, so that we found neurons that respond more intensively for the first, second, third, and so on elements of the sequential order of our task. Importantly, the population of tuned cells showed time-varying activity that was related with how the brain processes temporal and sequential information in our task. And here you can see evolving patterns of neural activity where single neurons are passing information to the next series of neurons across all our tasks. To try to understand these dynamics, what we did is to use multidimensional reduction PCA techniques to project the time-varying population neural activity into a low-dimensional state space. That allows to see how the brain is representing the beat. And what we saw is that the neural trajectories were cyclical, with each produced interval in the rhythmic sequence represented as a circular loop in these state-based cyclical neural trajectories. Crucially, longer intervals produce larger state trajectory loops, and this is evident across different intervals. In fact, the radius of neural trajectories increase monotonically as a function of the target interval, and in contrast, the speed of these neural trajectories tend to be constant. Importantly, the neural trajectories behave as periodic tangent circles with different diameters but converging in a state space attractor that is around here. In fact, we measure the distance of a constant point, here in red, and the position of the trajectories when the tap occurs. And we saw a constant distance relationship across target intervals, suggesting a very strong attractor behavior. In contrast, there is an increase in the distance for the half inter-tap points in the trajectory, suggesting that the amplitude of these trajectories is representing the dual time between taps. Some time ago, with my friend Hendjan Hunning from the University of Amsterdam, we suggested the gradual audiomotor evolution hypothesis that basically suggests that beat perception and entrainment emerge gradually in the primate order, with very large human flexibility to entrain to complex musical beats, but where monkeys have all the neural machinery to perceive and entrain to simple beats that are regular metronomes. So we have recorded then and compared now the neural trajectories using auditory versus visual metronomes with short and long tempos. You can see again that these neural trajectories are circular, with each loop representing one produced interval, and you can see that the auditory and visual conditions have different representations within the state space of population neural dynamics. You can also see that there is again an attractor behavior where these green points represent the tapping times. So for all conditions, the tapping occurs at a particular place in the trajectory, suggesting that the neural trajectories are representing the internal beat as an attractor in a particular state-space position. Here I'm showing the manifolds for duration, tapping movement, and the metronome modality, suggesting that these three processes are represented in the neural activity of supplementary and pre-supplementary motor neurons. Interestingly, now we are trying to understand how the bottom-up and top-down signals are interacting within the auditory, parietal, and premotor systems. And we are interested in all kinds of different processes related with this communication between the sensory areas and the predictive motor areas. So for this then, what we are doing is conducting multi-site simultaneous recordings in the behaving monkey using high-density silicon probes in the core and belt areas of the posterior stream of auditory processing, as well as in the pre-SMA and SMA areas, as well as the primary motor and the putamen in the basal ganglia, and the posterior parietal cortex. All of these simultaneously, while the monkeys are performing our synchronization task. Importantly, with these experiments, we aim to determine the passive auditory signals related with passive sensation, the tapping movement encoders, the active sensory encoders related with the perception of the beat, the internal beat signals, and of course the air encoded. So what I'm showing now are rasters of neurons recorded in different areas. So in this case, this is a neuron from the auditory core area, and you can see that this neuron has a very short and brief increase in activity after each stimulus during the perception, that is the orange dots, but also during the synchronization. And this response is very stereotypic, and you can see it also when the monkey is in a passive situation where it's not engaged in the task, since we remove the reward tube as well as the key and button elements. So the monkey is just listening, but is not really using this information. You can see that this auditory neuron has very similar responses during the task and outside the task. However, in the belt area, we also saw neurons that show modulation between the perception and the synchronization phases, and these responses were very different with low magnitudes during the passive task, suggesting that there is attentional responses in the belt areas. More interestingly, we also in the belt area saw neurons that were aligned not to the sensory events, but to the tapping events, and they have responses that occur particularly close to each tap, suggesting that the belt area has top-down access to the beat, internal beat signal. And these responses are completely gone when we test the neuron in the passive situation. We also saw neurons that represent the internal pulse in the pre-SMA and SMA areas, with clear responses aligned to each of the taps in the synchronization task, but do not respond during the perception. And also we saw very complex types of responses that are tuned to the duration or the serial order elements of the synchronization task. So these preliminary data suggest that there is a close interaction between the auditory and the premotor system during synchronization. And this interaction is very dynamic and depends on how the brain processes sensory information, but more importantly, how it predicts the temporality of rhythmic sequences. So I would like to especially thank Yaneri Ayala, Germán Mendoza, and Jorge Gámez for the development of all these projects. Gracias por su atención.